Hello students, a warm welcome to all of you in ITLS Academy and also welcome to all in our 3 days online workshop on topic homology modeling. In today's lecture we will discuss about the introduction of homology and we understand the overall idea of homology, what is the homology and what is the algorithm behind the homology. Okay, so we start today's lecture. First, we understand what is a protein. What is a protein? Protein is a structural and functional unit of cell. Functional unit of cell. Okay. Amino acid are the building block of protein. Is the building block of protein. Okay. What is a protein? Protein is the structural and function unit of cells and the amino acids are the building block of proteins. Okay. There are proteins are present in four conformation. First is a primary protein. And second is a secondary protein. And next is a tertiary protein. And next is a quaternary protein. A quaternary protein. These are the four conformations in which proteins are present. Linear chain of amino acid is known as a primary structure of protein. Whenever in linear chain we introduce the beta sheets and alpha helix, then it is known as a secondary structure of protein. Whenever in beta sheets or alpha helix we add the turn coils, hydrophobic, hydrophilic, lots of interaction, then it is known as a tertiary protein. Whenever the two polypeptide chains are two or more polypeptide chains are combined with each other, then it is known as a quaternary structure of protein. Okay. In biological system, there are lots of types of proteins are present. Some proteins are known and some proteins are unknown. Some proteins structures are known by the help of experimental methods, and these experimental methods may be X-ray crystallography, NMR, X-ray crystallography, NMR and cryo-EM. Okay, and some proteins structures are not designed by the help of these experimental methods. Okay, Okay, some protein structures are not defined by the help of this method. Okay, so th- that's why to understand the structure of these unknown protein we use homology modeling. Okay, in biological system in biological system there are lots of proteins are available. The structure of proteins are described or determined by the help of x-ray crystallography, nmr and cryo-EM. But there are some proteins are also available in biological system which structures is not determined by the help of these experimental methods. Okay, the structure of those unknown protein we understand by the help of homology modeling. Okay, what is the key aspect of homology modeling? The key aspect of homology modeling is this key the key aspect of homology modeling is to understand or to describe the 3D and quaternary structure of protein. This is the key aim of homology modeling. Okay, protein structure prediction is the most important aspect in computational biology that aim to determine the 3D structure of protein. Homology modeling is one of the computational method of protein structure prediction. Okay, what is a homology modeling? Homology modeling is one of the computational method or bioinformatic tool that are used to predict the 3D structure of protein. As the name suggests, means homology. Homology means homologous region in biological molecule. Here the biological molecule is protein. Or in protein sequence. Okay, protein sequence and protein structures are conserved. Okay, proteins have a what is a homology? Homology means a homologous region in a biological molecule, and here the biological molecules in protein. And in protein, we understand or we homology means homologous region in biological molecule. Here the biological molecule is protein. Okay, in protein we understand the conserved region of proteins. On the basic idea, the basic principle of homology modeling is we understand or we design the model of 3D protein with the help of homologous region in a protein sequence. Okay, suppose this is a sequence of amino acid. Okay, and the structure of this amino acid is unknown, and we want to predict the 3D structure of this unknown protein. Then in homology modeling we first understand or predict the homologous sequence of this particular. We search the homologous protein sequence of this particular amino acid. Suppose we perform a blast and we find out the similar sequence, and the similarity of this sequence is MTIFI. Okay, this sequence have a similarity with this particular unknown structure of protein, and the structure of this known protein is known, the structure of this amino acid is defined by the experimental method or computational method, that is a AlphaFold method, and the structure of this particular amino acid is determined by the experimental method, and the protein structure of this amino acid is defined by the experimental method or computational method. Okay, now this we use this structure as a template to understand the structure of this unknown protein. This is the principle behind the homology model. What is the basic principle behind the homology modeling? The basic principle behind the homology is the conserved or homologous region in protein sequences. First, if we want to perform the homology modeling of unknown protein, then first we find out the similar sequence, then we predict the structure of that homologous or similar sequence. To perform the homology modeling of unknown protein, first we understand or we predict the similar sequence. After predicting this similar sequence, we predict the structure of this known protein. Then we use this structure of protein as a template to model the structure of unknown protein. This is the principle behind the homology modeling. Okay, what is the homology modeling? Homology modeling is also known as a comparative modeling, because in this model we compare the known or unknown protein. Okay, homology modeling is known as a comparative. Homology modeling is also known as a comparative modeling because we compare the unknown or known protein structure to define the structure of unknown protein. Okay, homology modeling is also known as a comparative modeling. It is a computational method used to predict the 3D structure of protein based on an amino acid sequence or homologous amino acid sequence and the known structure of the relative protein. Homology modeling is a powerful tool in structural biology. Okay, it is widely used when the experimentally defined structures is unavailable. What is the homology modeling? Homology modeling is also known as a comparative modeling. It is a method to predict the 3D structure of protein with unknown structure by using a known structure of homologous protein. Okay, homology modeling relies on the identification of one or more unknown protein structure likely resemble to our query sequence. Query sequences are the unknown sequences, and on the production of sequence alignment that map residue in the query sequence to residue in the template sequences. By the help of sequence alignment methods such as BLAST, we align the template sequence as well as query sequences. Template sequence is the is the linear chain of amino acid of known protein, and query sequence is the linear chain of amino acid of unknown protein. Okay, what is a template? Template is a structure or sequences of known protein. Query means a structure and sequence of unknown protein. Okay. Okay. Proteins structures are more conserved than protein sequence among the homologous. These protein structures are more conserved. Proteins are more conserved. That's why we use these known protein as a template to understand the structure of unknown protein, because these known proteins or unknown proteins are the homologous proteins, and homologous proteins are more conserved, and the structure of homologous proteins are conserved. That's why we use or we design the model of that's why we easily predict the structure of unknown protein by the help of known proteins, because these proteins are conserved. Okay, and the proteins falling below the 20% sequence identity have very different structures. Okay, if the structure of protein sequences of two proteins have a similarity below to the 20%, then the structure of these proteins are different. But two proteins have a similarity more than 30% and more than 20%, then the structure of these proteins are very similar. Okay, and this is the principle, or this is the idea behind the homology modeling. Okay, now evolution. The quality of homology modeling is depend on the sequence alignment and template structure. Okay, the approach can be complicated by the presence of alignment gap. This alignment gap indicates that this structural region present in the target but not present in a template. Reason of the model that are constructed without the template are constructed by the help of loop modeling, and the reason designed by the loop modeling are generally much less accurate than the reason that are designed by the help of template protein or template. The reason of the model that are constructed without the template are designed by the help of loop modeling, and the structure and the reason designed by the loop modeling is less accurate. Okay, and the reason constructed by the help of template protein is more accurate, and the reason designed by the loop modeling is less accurate. Okay, now error in side chain packing error in side chain packing and position also increase with decrease the identity, and variation in these packaging configuration have a suggested as a major reason for poor modeling quality at low identity. Okay, side chain packaging and position also increase error in side chain and position is increases when the identity between the template sequences and query sequences decreases. Okay, identity and variation will decrease whenever the variation is increases and identity is decreases. Okay, this variation in this packaging configuration has been suggested the major reason of poor model or less identity. Okay, whenever the identity is increases and variation is decreases, then most probably poor model are designed. Whenever the whenever the identity is decreases, whenever whenever the whenever the identity is decreases and variation is increases, then most of the time poor model are designed. Taken together, these variation atomic position errors are significantly affect the use of homology modeling for purpose that require the atomic resolution data such as drug designing, protein-protein interactions prediction, quaternary structure prediction, etc. We understand the importance of homologous modeling. First is prediction of protein structure. By the help of homology modeling, we easily predict the 3D structure of unknown protein by the help of known protein. Okay, sometimes many proteins are lack experimentally determined structure due to the complexity and cost of the experimental method, and these experimental may be X-ray crystallography and cryo-electron microscopy. Homology modeling provides a reliable way to predict the structure of protein with unknown protein if the suitable template is available. Whenever a suitable template is available, then by the help of this template, we easily design the structure of unknown protein. Next, drug designing. In drug designing, homology modeling plays a most important role. Structure information from homologous model help in structure-based drug designing. Why identify the active site and designing model that can interact with target protein. Okay. In case of virtual screening, we potentially drug candidate are tested computationally against the protein target. Next, help to understand the drug resist mechanism easily to predict the evolving pathogens. By the help, next we understand the protein structure, function, and evolution. Okay, providing insight into how the protein is structured influence its function. By the help of homology modeling, we easily understand the structure of protein and we easily understand how the protein structure will influence the function of a particular protein. By the help of homology modeling, we easily understand the evolutionary relationship between the protein by comparing the model. Homology modeling can produce a high quality of structural model when the target and template are closely related to each other, and it plays the most important role in structural proteomics and structural genomics. Okay, first, next important is structural insight. By the help of homology modeling, we easily understand the protein function, interactions, and stability. In drug designing, this homology modeling plays the most important role in virtual screening and molecular docking. Next, mutation analysis. By the help of homology modeling, we understand the genetic variation and disease mechanism. Next, alternative to experimental method, providing a structural model when the experimental or NMR data are unable. This is the importance of homology modeling. Okay, now we learn the basic step of homology modeling. These are the basic step of homology modeling. First step is identification of target sequences, then database search, then identification of template, then refinement and sequence alignment, then building a model framework, then adding the loop and sidechain, then model optimization, then model evolution. These are the main steps of homology modeling. Okay, now we understand these steps in detail. Identification and selection of template structure. The first step in this process is to identify and select the appropriate template structure from protein databases, that is a PDB. First step is to identify a appropriate template. To search the appropriate template, we use the protein structure database, and this protein structure database is PDB, that is a protein data bank. Okay, to identify appropriate template, we perform a pairwise sequence alignment, and for performing the pairwise sequence alignment, we use a bioinformatic tool, that is a BLAST. What is a BLAST? BLAST is a basic local alignment search tool. Okay, this basic local alignment search tool can be used to identify the protein with high sequence homologous to query protein in PDB. To identify appropriate template, we perform a pairwise sequence alignment. For performing the pairwise sequence alignment, we use a method of BLAST. What is a BLAST? BLAST is a basic local alignment search tool. This BLAST can be used to identify the protein with high sequence homology with query sequences or query protein in the PDB. Okay, this BLAST is used to find out the homologous sequences to query protein in the PDB. Sensitive method involve performing a multiple sequence alignment or using an automatic iterative such as PSI BLAST. More sensitive method involving a multiple sequence alignment. For performing the multiple sequence alignment, we use a PSI BLAST. Okay. If the homology between the protein of interest and protein potential template is low or below the 35%, then we use the alternative methods, and these alternative methods are profile alignment and hidden Markov model. These hidden Markov model and profile alignment is used to reduce the gap in the alignment. Okay, once a several template is identified, then appropriate one used for the model. And the factor such as sequence identity, similarity, alignment score, phylogenetic relationship should be considered as well as other information such as biological function and environmental context. Okay. First step is identification and selection of template. Okay, to identify and select the appropriate template, we use first step is identification and selection of template structure. For identify and selecting the appropriate template, we use a protein structural database, and this protein structural database is PDB. Okay, to find out the appropriate template, we use a pairwise sequence alignment method. And this pairwise sequence alignment method is BLAST. What is the BLAST? BLAST is a basic local alignment search tool. And by the help of BLAST, we easily find out the homologous protein sequences. Okay, we easily find out the protein with higher sequence homology to the query sequences in the protein database. Okay, and whenever we perform the multiple sequence alignment, then we use the PSI BLAST. Okay, and whenever the homology between the template sequences and query sequences below to the 30%, then we use the another method, and these methods are hidden Markov model and profile alignment. And these methods are used to reduce the gap. Okay. Now we understand the evaluating template quality. First is the sequence identity and similarity. Next, evalue and bit score. Next, alignment coverage. Next, resolution of template structure, experimental method, and biological relevance. First, sequence identity and similarity. High value or more than 30% sequence identity give a best or high accurate model. Sequence identity and similarity. Higher value indicate the better template. Means whenever the identity and similarity more than 35% or more similarity have a more accurate structure. Next, e-value and bit score. Lower the e-value, higher the bit score indicate the better match, and we easily design a better or more accurate model. Next, alignment coverage, the percentage of target sequence covered by the template. More area are covered, more accurate model are designed. Next, lower the resolution is preferred, better structural detail. Okay, whenever the resolution is better, we understand the better structural information and we easily design a better model of our target unknown protein. Next, experimental method. X-ray crystallography and cryostructure methods are generally more reliable than the NMR. Next, biological relevance. Check if the template represents the correct functional and structural state of the protein. First is the sequence identity and similarity. More the sequence identity and similarity, better the modular design. Lower the e-value, high beta score, and better match and more accurate modular design. High area, high alignment coverage, better similarity and accurate modular design. Lower the resolution, better the structural detail and better the accurate modular design. X-ray crystallography and the template structure are designed by the X-ray crystallography and electron microscopy give the more accurate structure of unknown protein. Okay. The factors which affect the model, these are the factors which affect the quality of model. Next is alignment of query sequence with template. In homology modeling, the alignment of query sequence with template is an important step or important process. It is essential to achieve a high quality sequence alignment as it directly impacts the accuracy of the resulting model. Okay, a homology model can only be as good as the sequence alignment. Sequence alignment plays a most important role in sequence alignment plays a most important role in homology modeling. Better the sequence alignment, better the modular design. Okay. Multiple sequence alignment is preferred to contain a evolutionary information that is important for the structural and function. Sometimes we use a multiple sequence alignment. This multiple sequence alignment is more. Multiple sequence alignment is preferred to contain a evolutionary information. These evolutionary information is important for structure and function of protein. Okay, effective multiple sequence alignment. For performing the multiple sequence alignment, we use the proline and decoffee we use to perform a multiple sequence alignment. Okay. However, even the best program may contain errors, so it is necessary to visualize, examine the alignment to confirm that conserved residue are aligned correctly. Okay. In case of inaccuracy, manual adjustment where necessary, including the respiratory gap, changing the substitution score, moving sequence segment to improve the alignment qualities. Okay. To avoid the inaccuracy, we sometimes we use a manual adjustment, and these manual adjustment includes the in case of inaccuracy, manual adjustment may be necessary, including the reposting gap, changing the substitution score, moving the sequence alignment to improve the alignment quantities. Okay. For performing the better alignment, sometimes we use a manual adjustment, and these manual adjustment included the reposting gap, changing substitution score, and moving sequence alignment to improve the alignment quality. Sequence alignment plays the most important role to design a high quality of model. Okay, better the sequence alignment, better the model or design. Okay, sometimes we use a multiple sequence alignment. For performing the multiple sequence alignment, we use a proline, T-Coffee, and cluster omega. Okay, these are the bioinformatics tools that are used to perform a multiple sequence alignment. Okay, now we understand the third step, that is the building of three-dimensional model of query protein. Okay, sequence alignment is followed by the 3D model building of target protein. Okay, there are different methods used for generating the 3D model of protein based on a template. Okay, basically we divide or classify this method in 4 categories. First category is rigid body assembly. Next is segment matches, spatial restraint, and artificial evolutionary methods. These are the four methods that are used to generate the 3D model of query protein. First is the rigid body assembly. In rigid body assembly, the protein structure is broken down into the basic conserved core region, loop, and side chain. Okay, basically in this method we broke down the template protein structure into its basic conserved core region, loops, and side chain. These rigid parts are picked up from the template protein and brought together using a tool like 3D jigsaw, builder, and Swiss model. Okay. In rigid body assembly, first we break down the protein structure. When after breaking the structure, we pick up these body part of the protein structure, then design the model. Jigsaw, builder, and Swiss model are the bioinformatics tools that are used this concept to perform a homology modeling. Next is segment matching. Segment matching involves using a cluster of atomic position from the template structure as leading position, and these atomic positions are used to select the segment from the known structure in the databases on the sequence identity, geometry, and energy. These segments aligned with template structure to create a model of target protein. This is done by the sigmod and InCAD. In segment matching, we involve the cluster of atomic position of the template structure. In rigid body, basically we break down the protein structure. In segment matching, we in segment matching, basically the atomic position clusters are involved in segment matchings. In segment matching, we use the cluster of atomic position from the template query structure to design the 3D model of query template. Okay, these atomic positions are used to select the segment from the known structure in the databases on the sequence similarity geometry. And next is the spatial restraint method. In this method, we build the protein structure by applying the spatial restraint to guide the building of model that has closely matched the template structure. These restraints are determined by the stereochemical principle on bond length, bond angle, and dihedral angle, van der Waals contraction between the Control, van der Waals contact distance. Modular Modular is a software packaging commonly used to perform this method. Okay, and next is the spatial restraint method. In this method, we build the protein structure by apply the spatial restraint to guide the building of model that are closely match the template structure. These restraints are determined by the stereochemical principle on bond angle, dihedral angle, van der Waals contact distance. Model is a software package commonly used this method to perform a homology modeling. In this to build up the model, in this method we build up the model by applying the spatial restraint. Okay, next artificial evolutionary method. Artificial evolution method involves using the rigid body assembly method and set up the template evolutionary mutation together until the template sequence is matched as the target sequence. And this is performed with NEST. For building a 3D model of queries, for building a 3D model of query protein, we use a 4 method. First is the rigid body method, segment matching, spatial restraint, and artificial evolution. In rigid body assembly, we break down the template protein to design a 3D model of the query sequence. In segment matching, we use a cluster of atomic position to build up the protein structure. Okay, next is the spatial restraint method. In this method, we build up the protein structure by applying the spatial restraint, and these spatial restraints are used to guide the building of protein structure. And these restraints reason are determined by stereochemical principle of bond length, bond angle, dihedral angle, and van der Waals interaction. Next is the artificial evolution method. In this method, first we apply the rigid body assembly, then set up the template evolution mutation until the template sequences is same as the target sequences. Okay, so next we understand the loop modeling. Okay, there are often gaps or insertion that can occur in sequence alignment whenever we perform a modeling. Okay, and these gaps are called the loop. Due to this loop, the structures are less conserved during the evolution. These loops plays a most important role in protein structure. Loops modeling is important step in protein structure prediction because loops plays a crucial role in determining the function of protein, however predicting their structure in a complex process. Okay, there are two methods that are used to predict the loop. First is the data search method, next is the conformational method. First is the data search method. In data search method, we involve the comparing the target protein sequence with all known protein structure to identify the segment that can used to model the loop. Okay, in data search, basically we perform a comparison between the all known or unknown proteins. Next is the Next method is conformational search. In conformational search approach involving the optimizing a scoring function to find the best possible loop conformation. These method generated many random loops searching for one or fit the target protein structures. This approach is also called the ab initio because it does not rely the pre-existing protein structural information. There are some programs are also available for loop modeling, and these programs are Afreed, Petra and Coda. And these programs are Afreed, Petra and Coda. These are the programs that are used to perform loop modeling. Okay, generated due to the gap and insertion and deletions, and these loops are these loop plays a most important role in protein structure predictions and also plays a most important role in determining the protein function. There are two methods that are used to predict the loop. First is the data search method, next is the conformational method. In data search method we determine the loop by the help of sequence matching, and in conformational method we determine the loop on the basis of scoring value. Okay, there are some softwares or the programs are also labeled to find out the loop modeling, and these programs are Afreed, Petra and Coda. Okay, next is a sidechain modeling. Sidechain modeling is a process to predicting the conformation of side chain of amino acid in a protein structure. This process is crucial to evaluating protein ligand and protein interactions. This is usually done by placing the side chain on to the backbone coordinate that derived from parent structure or from ab initio modular stimulation. Searching every possible conformation in sidechain is a computational time consuming and not effective, so most sidechain predicting the program using the preferred conformational called rotamers, and these rotamers are stored in rotamer library which are collected in preferred sidechain conformation ranged by their frequency and occurrence. Okay, different energy function and such strategies are used to select the most appropriate rotamer for each amino acid side chain based on the preferred protein structure and giving backbone coordination. There are some tools are also available to perform a sidechain modeling, and these are the tools ramp, and these are the bioinformatics tool that are used to perform a sidechain modeling. Sidechain modeling is very important to evaluating the protein structure and protein ligand structure. Through performing the rotamer library plays a most important role to building the sidechain of a target proteins. Okay, now model optimization. The next step is to optimize the model. Optimization of model refer the process to refine the initial model to improve the accuracy and reliability. To improve the reliability and accuracy of the model, we use the model optimization. This model optimization process adjusts the position of atom in the model to reduce their clashes and steric hindrance between the molecule. Energy minimization process is one of the method of model optimization. It calculates the potential energy of model, adjusts the position of atomics to minimize this energy. Excessive energy minimize can often more residue away from the correct position, so only limited energy minimize is recommended. Okay, in addition, the energy minimization optimization techniques such as molecular dynamics, Monte Carlo stimulation can be used. Molecular dynamic stimulates involve the stimulating the movement and interaction of atomic in model over the time, which allow the exploitation of different conformation to identify the most stable and energetically favorable one. In Monte Carlo stimulation, use the random sampling to explore the different configuration of molecule and identify the most stable and energetically favorable one. Okay, now we understand the model optimization. Model optimization is a one of the most important step in model in homology modeling. By the help of model optimization, we improve the accuracy and reliability of our model. Okay, in optimization process, we involve the adjustment of we adjust in model optimization, we adjust the position of model. Okay, and basically we reduce the clashes and steric hindrance. And we also in energy minimization is the most important step of model optimization process. Okay, there are two methods that are used to minimize the energy. First is a molecular dynamic method, next is a Monte Carlo stimulation method. Okay, in molecular dynamic stimulation, we involve the stimulation, the movement and the interactions between the atom over the time. Okay, in Monte Carlo stimulation, we use the random sampling to explore the different configuration of model to identify the most favorable reason. Okay, model energy optimization is the most important role in model optimization. Next is the model validation and evaluation. After 3D after 3D of after 3D model of query protein has been built, it is essential to validate and evaluate its quality, ensure the biological reliability, and can be used for further study. Okay, it involves the comparison. This involves comparing the model to real experimental 3D structure using a various evaluating models, stoichiometry, physical parameters, knowledge-based parameter, statistical machinery, and other criteria. Okay, some common used program for evaluating protein structures such as VITIF, VITIF, ProCheck, and PROSA. Okay, these programs compare the protein of homologous model to find the experimental structure in the PDB. The quality of the quality of homology modeling largely depend on the accuracy of sequence alignment and and the part of protein where the template is not similar to the query protein. Okay, model validation and evaluation plays a most important role. Okay, we in for validate the protein, we comparing the model to realistic 3D structure using the various evaluation parameters. And these evaluation parameters are the stoichiochemistry property of our design 3D model, physical parameters, knowledge based parameter, statistical mechanism, and other criteria. Also there are some bioinformatics tools are also available to validate our 3D model, and these tools are VITEF, ProCheck, and PROSA. In ProCheck, we understand the Ramachandran plot. Ramachandran plot is also a method to evaluate our 3D structure of unknown protein, which are designed by the help of homology modeling. These are the steps which are used in homology modeling. First step is a identification of template, then sequence alignment, then building the 3D structure, then optimization, then validation and evaluation. These are the methods which are used to perform a homology modeling. Okay, in this workshop we understand the homology. In this workshop we understand how we perform the homology modeling through SWISH model. Okay, this is the software which is used this is the software or web-based this is the web-based method which are used to perform a homology modeling. How we perform a homology modeling we discuss in next class. Okay, this is all about today's lecture. Please follow ITLS Academy on Instagram, Facebook, Twitter, YouTube, WhatsApp, LinkedIn, and also get it on Google Play Store. Thank you so much for watching my video. Please like, share, and subscribe ITLS Academy. Thank you.